Welcome to the lecture on genes and gene frequencies. The first part of this lecture was recorded a while back, just an FYI. So, to understand genetics, you have to go back to the gentleman who pioneered the field. Gregor Mendel is called the father of genetics, that's him. He was an Austrian monk who lived in an abbey with other monks, and they had a communal garden. And he was a trained scientist at the time, but he did some studies on these pea plants and ultimately came to explain the modern laws of genetic inheritance. And even though we now have a much better understanding of genes as a whole than Mendel ever would have had, his general concepts are still shown to be true today, much as they were in the 1800s. So it's pretty cool because a lot of science from 150 years ago we've since improved upon to tremendous degrees, but Mendel's basic ideas about genetic inheritance, they're still taught today, as you can see, so it's pretty cool. Genetics, study of heredity. Heredity, transmission of traits from one generation to another. Mendel wanted to explore how organisms passed on their genes. It's not, I ate some peanuts and my throat is dry. That's all it is. So, genetics is a study of heredity, and heredity is the transmission of traits from one generation to another. When Mendel was alive, one of the dominant ideas was the blending hypothesis. This taught that basically mom was red paint, dad was white paint, their child would be pink paint. You are a mishmash of the sperm and egg cell from your parents, ultimately passed on traits from both, but you are just a hodgepodge mix of both of them. The thing that the blending hypothesis did not teach was that genes, when passed from mother and father to the offspring, retained their uniqueness when they came together to make the child. Let's say you have red hair. Your parents don't, your grandparents don't, but your great-grandmother does, or did. Same hair, when she was younger, she had red hair just like you did. So, how did that trait skip a generation, 2 generations actually, and show up in you completely unchanged from what it was 2 generations ago, when your great-grandmother had the same hair color? Blending hypothesis did not explain this. Blending hypothesis would have taught that the red hair color was gone, blended out, and yet it showed up again, unchanged, after skipping generations. This was an insufficient hypothesis for how genetic inheritance worked. Mendel knew that, he wanted to explain how it worked, and he did. Ultimately, through his pea plant experiments. So let's get to those. There is a pea plant flower. Mendel, by the time this was done, he actually he published an article about it. By the time he published his article, he determined that individual organisms pass on distinct, he called them heritable factors. We now know them as genes. And that these genes were passed on, and they did not become a mishmash, they retained their uniqueness when passed from parent to offspring. How does he figure this out without any of the modern equipment that we have? Well, let's talk about how he did. He figured it out by starting with some controlled breeding. So here's some pea plants. You got purple and white flowers. Mendel, first of all, made sure the plants did not self-pollinate. So if you remember from Flowering Plants Lab, which we did, that was the last lab that we did. If you remember that, pollen from one plant needs to enter the carpel and fertilize the ovule of the other plant. Plants are, most of them, monoecious. It means they are both male and female structures for reproduction. And so Mendel cut off the stamens of the purple flowers, used a paintbrush or other equivalent device to transfer pollen from the white flowers to the purple flowers, and thus artificially pollinated them. This guaranteed that he knew which plants were the two parental plants. They produced peas, he took the pea seeds, planted them, and take a look at all of the offspring made by that union. All of them are purple, every single one. So, Mendel first needed parental plants, one purple flower, one white flower. Together they made the parental generation, or the pea generation. That's these two up here. All of the offspring produced by that union would be the F1 offspring. And if you look at them all, every single one is purple. Blending hypothesis would have taught us that purple was the dominant physical trait, and the white coloration was blended out after simply one generation. It's gone, it's blended out. If these guys were to pollinate each other and produce seeds, the next generation and the one after that should continue to show purple coloration. The white color is gone. That's what the blending hypothesis would have said. Okay? So, parental generation, purple and white, offspring produced by that, all purple, they're called the F1 generation. This next part harkens back to the previous chapter when we talked about mitosis and meiosis. We talked about cell division. Do you guys remember the word diploid? Hope you do. What it means is two copies of each chromosome. If a cell or an organism is diploid, it has two copies of each chromosome. In an organism, such as yourself or a pea plant, you have mostly what are called somatic cells. These are all of your cells other than egg cells or sperm cells used for sexual reproduction. Those are not diploid. We'll talk about what they are in a little bit. But all body cells, all somatic cells are diploid, two copies of each chromosome. For instance, again, it's not this simple, but just for illustrative purposes, eye color. If there were a single gene that controlled eye color, you have two copies of it, one from each parent. Your dad would have given you one gene for eye color, and your mom gave you one, and now you have a pair of that gene. You have two copies of it. So, you're diploid. The flowers are diploid too. There's our flowers. Purple one, white one. These X-shaped structures are chromosomes, right? And notice how purple flower has this particular part on the chromosome that codes for a particular gene for flower color. So, on one particular chromosome, you've got 23 pairs of chromosomes. I don't know how many pairs pea plants have, but on one particular pair of them, there's going to be a section that has the gene for flower color. Since the flower itself is diploid, it has two copies of that gene for flower color. It's got the copy here, it's got the copy here. Each flower has two copies that code of this gene that code for the color of the flower. The white flower has two, purple flower has two as well. Notice how I've used different colors to represent the genes for flower color in the two different colors of pea plant. That's because they have different versions of that particular gene. Flower color is a character. A character is a feature, a heritable feature. That means it's passed on from one generation to the next. A heritable feature that varies among individuals. Well, you can't look around you because we're not in a classroom, but if we were in this classroom, we'd all look different. There's obviously a number of physical features that are different among us. Hair color would be one, eye color would be another, height would be another. These are all characters. They're controlled by our genes, and they vary among a population of individuals. In our pea plants, there's a lot of different characters. One such character would be the color of the flowers. there are multiple versions for most genes. Think about hair color. There are several different colors, naturally occurring colors of hair. Several versions of that gene, and in this case, hair color, it's a lot of different genes. But for our pea plant flowers, there's only two versions in this case, purple and white. When you're looking at things in genetics, oftentimes if there's only two versions of a gene, an English letter is used to represent the two different versions. In this case, the letter P is used. We use letters when there's two different versions of a gene because it's pretty easy to have two different versions of a letter, capital and lowercase. In pea plants, coloration, purple or white, is called a simple. It's a simple dominance inheritance pattern, which means that there is a dominant version of the gene and a recessive version of the gene. If they occur together, you're going to see the dominant one. And in this example, in Mendel's experiment, purple petals, purple flowers, are the dominant trait. White flowers are the recessive trait. So we use capital P to represent a copy of the gene for being purple, having purple flowers, and a lowercase p to represent the gene for white flowers. So let's go back then. Let's go back a slide. Our purple flower, we said that it has two copies of the gene for flower color. Here's one, here's two. Both of them are colored purple, which means both copies of the gene for flower color for our purple pea plant, say that 10 times fast, would be big P, the purple gene. Our white pea plant has two copies of the lowercase p, the white color allele. You haven't heard that word yet. The white color gene. So we've got purple plant, two copies of the purple gene. White flower, white plant, two copies of the white color gene. Big P, little p, little p. Okay. Mendel used those two plants as his P generation, his parental generation. He mated them together. If you remember how human chromosomal inheritance works, each parent passes on one of their two copies of a particular chromosome. So if these two pea plants were going to reproduce together, one copy of the chromosome will be passed on to the offspring, the other one won't. Same thing for the white flower. And which one gets passed on? Random chance. The offspring then are going to get one of each letter from each parent. From the big parent, or the big P, purple parent, sorry. It's going to get a copy of the purple gene. That's all that that parent can give. The white flower, the only gene it can pass on is the lowercase p, which means all of the offspring produced by this union will have one copy of each type of gene. Okay. Move on here. So here's the word that I used a little while ago that we hadn't defined yet. The word is allele. An allele is an alternate version of a gene. So alternative versions of a gene are called alleles. All right? In this example, purple allele and white allele. In Mendel's P generation, the purple flower had 2 purple alleles, while the white flower had 2 white alleles. Big P, little p, little p. Another word to note is the word homozygous, right? Homo means same, so in this case it means same alleles. The purple flower is homozygous dominant because it has the dominant physical trait, the dominant allele. The little p of the white flower is homozygous recessive because it has 2 copies of the same recessive allele. Both homozygous, but homozygous dominant, homozygous recessive. Punnett squares. You've probably made these at some point. Punnett squares show us all possible offspring that can result from the union of 2 parents. And let me look at my phone and make sure, A, that it's still recording, which it is, and it looks like I got more space on the left side. So we're going to make a Punnett square over here. Punnett squares show all possible offspring that can result from the union of 2 parents. Unfortunately, I do not have a purple marker. That's okay. So, rule number 1, I want you to remember this. If there's one English letter, then the Punnett square is going to have 4 total squares, 4 possible offspring. All right? If there's 2 English letters, it gets a little more fuzzy. So we'll get to that later. So 2 parents, when you're making a Punnett square, go above and to the left of the Punnett squares themselves. A little bit better. So these are the possible offspring. To the left and above the columns, that's where you're going to have possible gametes. And I'm illustrating here with sperm cells and egg cells to really hit the point home. The male, or the functioning as a male parent flower, right? We're going to say that that was the white flower. The alleles it had were white and white. And we'll use blue to represent the purple allele. So our other parent in this union had 2 copies of the purple gene. So parent number 2 can pass on either the first white allele, that's one possible sperm cell, or the second white allele, that's the other possible sperm cell. Parent number 1 can pass on the first purple allele or the second purple allele. Those are possible egg cells. So then you fill it out like a multiplication table. Big P with little p. And I think that's going to happen for all 4 possible offspring. Every single one is going to be big P, little p. That is how Punnett squares work. They show you all the possible offspring produced by the union of 2 particular parents. In Mendel's experiment, he got these offspring, and all of them collectively were called the F1 generation, and they all looked purple. because complete dominance is how this works in pea plants, and purple is completely dominant over white coloration. So, all of them are purple. But notice that the white allele was hidden. It was masked. It was still there. You just didn't see it. That's why the blending hypothesis doesn't work. It didn't take into account the fact that these alleles were recessive and hidden for generations. Please note that this is called complete dominance. There's another thing called incomplete dominance when the recessive allele is not completely hidden. We'll get to that later. So homozygous describes both of the parents here having two copies of the same allele. Heterozygous is the term that describes all of the F1 offspring because they have two different alleles. Mendel decided to pair a couple of his F1 offspring together and see what happened. This would make the F2 generation. Let's go ahead and do that Punnett square up here and see what that makes. Both of the parents would have been heterozygous, big P, little p. So parent number 2 could have passed on a sperm cell with the purple gene, possibly, or it could pass on a sperm cell with the white gene. Same thing with parent number 1 over here. So now let's fill out this Punnett square. Offspring possibility number 1. It gets the purple gene from both parents. It is homozygous dominant and looks purple. Possibility number 2. It gets the purple gene from the male parent and the white gene from the female parent. It's heterozygous, but it still looks purple. Possibility number 3. Gets the white gene from the male parent, the purple gene from the female parent. It's heterozygous and still looks purple. And then possibility number 4, it receives the white gene from both parents and it looks white. That was the real kicker in this experiment for Mendel. It showed up, the white color showed up after skipping a generation, totally unscathed, exactly as it looked in his original P generation. It wasn't blended out, it just was hidden, it was masked. And so that was the major aspect of this that taught Mendel about genes and the uniqueness of genes and how they maintain their uniqueness when passed from one generation to the next. All of these offspring, by the way, would have been Mendel's F2 generation. Couple of terms to note: genotype versus phenotype. Phenotype is an organism's physically expressed trait. Genotype is an organism's actual genetic makeup. And here's a way to really distinguish them. These two hypothetical pea plants have the same phenotype, right? They're both purple. However, genetically, they're different. This one's heterozygous, big P, little p. This one's homozygous dominant. Different genotypes, same phenotype. So keep in mind the difference between those. Genotype's the actual genetic makeup. Phenotype is what it physically looks like. Here's another good way to help you understand with the difference between phenotype and genotype. If an organism shows the recessive phenotype, if there's a white pea plant, it has to be homozygous recessive in genotype. For instance, in the pea plant example, it's little p, little p. However, if something shows the dominant phenotype, purple flowers, you have no way to know just by looking at it whether it's homozygous dominant or heterozygous. So how can you tell? You can perform what's called a test cross. I'm not sure if you can see the picture too well, but that's a black lab, and that one is a chocolate lab. I doubt you can see the fact that it's brown very well, but that's okay. Let's talk about labs and lab fur color. There are 3 principal varieties of Labrador Retriever fur color, and for at least two of them, it's a very simple genetic component that causes the fur color. So, in labs, big B, dominant allele, codes for black fur. Lowercase b, recessive allele, codes for brown or chocolate fur. Just like our pea plants, each dog is diploid, which means each dog has 2 copies of the gene for fur color. Yellow labs, it's a whole other gene that causes that. So we're not going to worry about yellow labs. This is the black lab up here. Chocolate lab is down here. If you look at a chocolate lab, you know, based on its phenotype, brown fur, what its genotype has to be. The only way you're going to have brown fur is if you have 2 copies of the recessive gene, the recessive allele, little b, little b. But if you're a black lab, you could be homozygous dominant or heterozygous. So if you've got a pet black lab here, how can you know which of these your dog is? One possible way is to look at the parentage. Right? If the parent of this black lab was a chocolate lab, then you know it had to get one of the brown genes from that parent. But what if you don't know the parentage? If you don't know parentage, one way to do this could be a test cross. If you have a black lab, please don't actually do this. There are enough puppies that need adopting without doing a scientific experiment right now to produce additional puppies. But this is how you could do it, hypothetically. One English letter, the letter B, so it's a 4-square Punnett square. Your black lab, we'll put over here. You're going to want to mate a black lab with a chocolate lab and see what the puppies look like. Let's say that your black lab is homozygous dominant. If you mate that black lab with a chocolate lab, every single puppy will be a black lab, because the only gene that your black lab can pass on is the dominant one. On the other hand, if your black lab is heterozygous, then roughly half of the puppies should be chocolate labs. And in fact, if you get even just one puppy that's a chocolate lab, you know your black lab must be heterozygous. And that's called a test cross. It's a way of testing the dominant phenotype to see which genotype it actually is. All right. This next slide summarizes Mendel's paper that he published regarding patterns of inheritance. So I'm just going to read through these real quick. Number 1, Mendel noted that alternative versions of genes, which we now call alleles, account for variations in inherited characters. And if you hear a strange sound, it's starting to pour outside, so that's what you're hearing. So alternative versions of genes account for variations. Just look around. If we were in the classroom, look around the room, and you'd see a lot of variation. For each character, so for each physical feature that an organism has that's controlled by a gene, they inherit 2 alleles, one from each parent, because living things generally are diploid. If the 2 alleles of an inherited pair differ, so heterozygous, then one determines the organism's appearance and is called the dominant allele, the other has no effect and is called the recessive allele. This is true for traits that involve complete dominance. There are some traits where recessive alleles are not totally masked. Mendel's last statement, his last hypothesis, a sperm or egg cell carries only one allele for each inherited character because allele pairs separate from each other during the production of gametes. So again, you've got, this is just hypothetical, it's not actually this simple. You could have 2 alleles for eye color. You could have one brown, one for blue. You would pass on one of those to your children. And if you have more than one kid, then it's a random chance each time. You might pass on the brown gene for brown eyes to one child and the gene for blue eyes to the other child. Again, eye color is not that simple, but if it was, that's how it would work. Mendel stated this, and this is true for complete dominance. It's not true for all traits. Some traits do not involve complete dominance. In humans, the type of simplicity that we see with our chocolate lab example and our pea plant example, we don't usually see it in humans. Most human traits are controlled by many genes. Good, still recording. So, it's called Mendelian genetics, involves complete dominance and physical traits controlled by single genes. This is not common in humans. But we'll look at a couple of genes in humans that are controlled by one gene, including albinism. This is an albino squirrel. To be albino means to lack all melanin, no pigment. The key here is you look at the eyes. If the eyes are pinkish or reddish, then the organism is albino. There are albino humans, but it takes 2 copies of the gene to show it because being albino is recessive, right? And 1 copy for normal pigmentation, you will show normal skin pigmentation. Sickle cell anemia is caused, or sickle cell trait, I should say. That'd be a better way to put it. Anemia is the disease. Or I should say anemia is a symptom caused by sickle cell trait or sickle cell disease. But sickle cell trait is controlled by a single gene. Cystic fibrosis can be, and achondroplasia can be as well. That's Peter Dinklage. He's probably the most famous actor with achondroplasia. So it's a type of dwarfism, but it's different from these other traits in that it is a dominant allele. So if an individual has 1 copy of each, the recessive is normal height. The dominant allele is achondroplasia dwarfism. So it's unusual for a lot of these single cell traits to have the condition be the dominant allele. Part of the reason why it is for this one is because achondroplasia it's not a harmful condition to have. So, most traits in humans, polygenic. Polygenic means multiple genes. Most human phenotypic characteristics are polygenic. A couple examples, eye color and hair color. So I've used eye color as a simple example here a couple times, but it's not that simple. Eye color is controlled by multiple genes. So is hair color. So is your height. So is your skin color. So is a lot of stuff. Most human things. There's a picture of eyes, and here's at least 2 of the genes we've isolated that we know influence eye color. You do not have to memorize any of that. Any of this. So, Mendel looked at complete dominance. There's also incomplete dominance. Incomplete dominance is when one allele does not completely mask the other one. So, an organism that's heterozygous is going to have an intermediate physical characteristic. Snapdragon flower color is a good example of it, but I prefer hypercholesterolemia. Hyper means above, cholesterol means cholesterol, and emia means blood condition. So hypercholesterolemia means high blood cholesterol. High blood cholesterol levels. So, if you've got no copies of the gene, okay, if you have 0 copies of the gene for this cholesterol addition, and actually I wrote that wrong. There you go. There we go. If you have 2 copies of the normal gene, you have normal blood cholesterol. So the big A is normal. So if you have 2 copies of that gene, you have normal blood cholesterol. If you have 2 copies of the gene for elevated cholesterol in the blood, you have very high blood cholesterol levels. And I hope this is all showing up on the screen. I remember that side of the screen had a little bit less coverage. Yep, it is. Awesome. So if you had 2 copies of the recessive gene, you'd have extremely high blood cholesterol levels. But if you've got 1 of each, your blood cholesterol is slightly elevated, potentially enough that you might need certain drugs to lower it, you need to watch what you eat, exercise, etc., but it's manageable. People who have 2 copies of this gene for hypercholesterolemia, doesn't matter what they do, their blood cholesterol is going to be through the roof, and they'll need medication for it. So this is an example of incomplete dominance. Codominance. This is fun. This is blood typing. So we're going to ignore the positive negative inheritance for this. We're just going to look at the lettering for blood types, A, B, and O. So in terms of the actual blood types, they're the phenotypes. There are 4 of them. A, B, AB, and O. I'm actually going to stop it here because I think I've gotten well past 30 minutes on this thing. And we'll record the other half of this in another video. All right, welcome back everybody. So, continuing our genetics lecture, we are at codominance, when 2 alleles, which are alternative versions of a particular gene, are equally expressed. It is called codominance, and human blood type works like this. The four blood phenotypes, ignoring positive and negative, are A, B, AB, and O. There are actually only 3 blood genotypes. There is the A genotype, the B genotype, and then the recessive genotype. Both the A and B genotype are dominant. The lowercase I that you see here is recessive. Now remember, each person is diploid. They have 2 copies of each gene. So, everybody will have some combination of 2 of those blood genotypes. Let's just put all the possibilities up here. I want to make sure that that is on camera. It is. Perfect. So these are all of the possible genotypes that you could have for blood type. What are they actually talking about, by the way? What does A and B even mean? Well, they refer to antigens that are present in your blood. Type A blood produces type A antigens. Type B blood produces type B antigens. The recessive allele codes for producing no antigens. What's an antigen? It's something that your body would ordinarily recognize as a foreign, potentially dangerous substance, and it would react accordingly. However, if you have the genes that code for the production of a certain type of blood antigen, your body knows that it's supposed to be there and it won't attack it. So type A blood, people with type A blood, they produce type A antigens. Type B blood produce type B antigens. And AB blood produces both. Let's just draw that. The way I'm going to draw these antigens is nowhere near accurate, so please know that. Type A antigens, we're going to represent with this symbol. So, somebody who has type A blood is going to have A antigens with their blood and recognized by their body as a normal thing. Type B antigens we're going to show as a little semicircle. And someone with type B blood would have type B antigens in their blood recognized by their body as a normal thing. People with AB blood have both. Their body recognizes both A and B antigens as being normal. Type O blood is somebody that has no antigen recognition. So if you have type O blood, your body doesn't recognize any of those antigens as being normal. So what if I'm type O? What if somebody with type A blood tried to donate blood to me? Well, my body does not genetically recognize type A antigens as being normal, and so my body would attack that foreign blood, and that reaction would ultimately, most likely, kill me. So I can only receive blood from other people who are type O, because their blood doesn't have any antigens either. On the other hand, if you're type AB blood, you can get blood from anybody, because your body recognizes all the antigens as being normal. How does this relate to genetics? Well, you've got 2 copies of each individual gene. And by the way, genotype usually refers to an individual. So this thing over here where I said blood genotypes, it's really much better stated as blood alleles, right? Versions of that gene. Genotype is in reference to an individual's combination of alleles. So if you took any notes on that, make sure to fix that. I apologize for that error. So here are the genotypes. Each of them is going to yield a particular blood type. I think you can probably figure out what they are. To be type O, like me, you have to have 2 copies of the recessive gene. To be type A, you simply need 1 copy of the allele that codes for type A antigens. Same story with B. If you've got both A and B, then you are type AB blood. And that's this codominance idea. It means that both A and B alleles are equally and fully expressed. It isn't some intermediate half A, half B thing. It's fully A, fully B, both shown, both expressed. And when they occur together, they're codominant alleles. So, blood type, codominance. So I want to revisit this term, phenotype. What is an organism's phenotype? Pheno, physical. An organism's phenotype is its observable physical characteristics. And phenotype ultimately isn't just caused by an organism's genes. It's caused by the genes, but also environmental factors that impact that organism. Only the genetic component is actually passed on to the offspring. So only the genetic component of an organism's phenotype is heritable. Certain traits are not heritable. For instance, if you have an accident and lose an arm, and you have children, you're not going to pass on that lost arm. That's not heritable. That's an environmental effect on your physical being, not something that is genetically linked. One challenge is to determine how much of an organism's physical being, phenotype, is actually caused by the genes and how much is caused by environmental factors. In other words, how much of it is heritable. I've got 2 pictures here. Let's start with the one on top. It's a picture of a giraffe. I put this up just so you're aware of the misconception that existed about inheritance prior to our understanding of genetics with Mendel. I'm not sure, I can't remember if I actually covered this in the previously recorded lecture, which you will have just watched. But in case I didn't, the name for the dominant thought at the time of, or time preceding Mendel in the 1800s was Lamarckism. This was the idea that when you were alive, you did stuff that impacted your physicality and you passed that on to your offspring. For instance, giraffes stretch their neck to reach high up into the trees. If a giraffe stretches its neck more than another giraffe, it will have offspring with longer necks than the other one that didn't stretch it as much during its lifetime. But we now know that's not how natural selection works. That's not the way it works. Instead, giraffes that genetically would have had the code for slightly longer necks than their peers, those would have experienced greater reproductive success, and thus, over time, you would have had that tendency toward the longer neck. It wasn't because they stretched it out that they ended up having offspring with longer necks. Down here at the bottom, we've got a picture of 2 identical twins. So genetically, these 2 are identical, 100% the same genes, and yet one of them is clearly taller than the other one. So that tells us that height is not entirely caused by genes. Your nutrition that you get when you are growing your environmental factors also play a part in determining how tall you're going to be. And if you look throughout human history, average height has shifted in response to environmental factors. For instance, bone studies indicate that during the medieval times, when it was relatively warmer, people actually grew an additional inch or two compared to a little bit later in the 1800s when it cooled down and there was slightly shorter growing seasons, and so the actual average height was about an inch or two shorter based on studies of bone fragments that people have done from that time. So for people, height is an example of a trait where you've got both genetic and environmental factors that influence it. Okay? This is kind of, if you go into nature versus nurture, that whole debate, that's pretty much what this is. So how do you figure it out? How do you determine how much of a particular physical feature is caused by genes versus environmental impact? The truth is, it's really, really hard to do. In fact, most of the time, it's nearly impossible. So heritability of a particular trait is defined as the proportion of the total physical phenotype variation in a trait that is attributed to the genes. So how heritable is something? That means how much is it genetically linked. If you want a human example to think of, let's say there's a certain type of cancer. Physicians, doctors might be interested in figuring out how much of this cancer risk is heritable. In other words, how much of it is outside of your lifestyle control. Or if it's not heritable, then it might be caused entirely by something that you're doing. For instance, lung cancer, they link almost all cases of lung cancer to either smoking, secondhand smoke, asbestos exposure, environmental stuff. Lung cancer seems to be mostly not heritable. But other cancers like breast cancer, testicular cancer, they don't seem to be linked to anything environmentally most of the time. It's just genetics. And so it seems to be potentially those cancers could be more heritable. They might just be bad luck mutations as well. But you get the idea, environmental versus heritable. So yeah, usually it's both, and usually you can't tell. There's one study that is often cited in ecology that shows how you can occasionally study this whole nature versus nurture, environment versus genes impact on phenotype. And it involves this little owl right here. It's called a Ural owl. It's where it lives in the Ural Mountains over in Europe. So that's what it looks like. They live in northern European forests. They eat these things called voles. They're these little funky looking rodents, kind of like mice, little stout mice without tails is kind of how they look, with very short tails. So little rodents, but these rodents are a boom and bust rodent. So basically there's either a ton of them and the owl can feast, or there's very few of them and the owl is going to have a hard time getting the food that it needs. Many owl species actually move in response to the voles. If there aren't enough voles to eat, a lot of owls will go south, come in the US here at least, they'll go from Canada coming into the US. In Europe, they would go further south into Europe as opposed to staying up in the northern parts. But most owls will move in response to voles. These guys don't, they just stick around. They stay and nest in the same spot even when the vole population is down to very, very little. So, one way to test heritability with these guys is they compare the size of the eggs, the number of eggs that get produced, and they compare for this owl from good years and bad years. So, if the owls produce the same clutch size, let's say they produce a clutch of 5 eggs. If they produce 5 eggs during good years and bad years in terms of food availability, then that means that egg size or clutch size is not influenced much by the environment, but is actually genetically coded. So if it's the same number of eggs produced in good years and bad years, then it's genetically linked. On the other hand, if they produce a clutch of about 8 eggs in good years and about 2 or 3 in bad years, then the number of eggs they make is influenced by environmental factors more than their genes. They found out it's mostly the genes. The clutch size doesn't really change no matter how much food there is. But this is one example of how you could study nature versus nurture, environment versus genes. But most of the time it's really hard to do it. The identical twin studies are a way that you can do this with people, right? Because identical twins have the same genes. So any difference between them is environmentally linked. All right. If anyone's a Harry Potter fan, you recognize that that's Ron Weasley. We'll get into why he's there in just a moment. Population genetics looks at the gene frequencies in the entire population. If you think back to what a population was all members of the same species in a particular area. So that can be, like for humans, right? The world population is just under 8 billion, I think now. The population in St. Louis is 300,000, 400,000, something like that. Population here in Rolla is 20,000. So depends on what you're looking at in terms of particular area. It's quite variable. But population genetics looks at genes distributed in a population of organisms. Mendel didn't focus on population genetics, but ultimately his work laid the foundation for it. And one area of interest with population genetics is the gene frequency idea. If you remember what an allele is, it's an alternative version of a gene. And so Mr. Weasley is here because we're going to talk about red hair as a particular allele. There's more than one mutation that ultimately leads to red hair, but considered as a whole, less than 2% of the world's population has red hair. We're going to look at one country in particular, the country with the most redheads in the world, which is Scotland. I guess it's not a country, it's part of England, but meh. So, Scotland has, from what I was reading, 13% of people with red hair. So this is the number that actually express the phenotype of having reddish colored hair. It's a recessive phenotype, so you don't actually show it unless you have 2 copies of the gene. In terms of the actual frequency of the red hair allele, estimates put it at roughly 40% of individuals in Scotland have the allele that causes red hair. Again, if you don't have 2 copies of it, you won't actually show it. But 40% of people, they estimate, have the allele, which was caused by a mutation sometime in the past, that causes red hair. So other countries, localities will have very different frequencies of that allele. This is the place in the world where it's the highest in occurrence. Also, if you want to tie this back to a non-human creature, we have the guppy study we talked about. So once you put the guppies in a predator-free stream, you no longer had the pressure selecting against the bright guppies, and so the frequency of the frequency of the allele for bright males increased once the predators were gone. This next part can be a little tricky for some people, so I want you to make sure you listen carefully to how I'm going to describe it. So at any one particular point in time, you have a certain distribution of alleles, a distribution of different versions of particular genes. For instance, at one time, 40% of individuals in Scotland have the gene for red hair. Over time, this ultimately changes, which is biological evolution, changes in gene frequencies over time. Using the guppy example, eventually we saw a shift toward a much greater number of individuals having the bright colored gene, with natural selection guiding it in that direction. The Hardy-Weinberg principle says that whatever the current distribution of genes is, whatever the current gene frequencies are, Hardy-Weinberg principle says that they're going to stay exactly as they are. It's not going to change. Evolution will not happen if certain conditions are met. So the Hardy-Weinberg principle says that gene frequencies will stay exactly the same. In other words, evolution, natural selection, none of that's going to happen, provided that certain conditions are met. And here are the conditions. Number one, mating is random, which means females don't pick males that have superior traits, or vice versa, or there's no sexual selection at all, it's just totally random mating. Condition number two, there isn't any movement of genes in or out of the population. There's no immigrating in or emigrating out of individuals. Mutations don't happen. Those are changes in the genetic code due to random chance. Large population size needs to be present so that we don't have genetic drift, which will be the next slide we'll talk about. That's a pretty vague statement, by the way. Large is relative. Also, equal fitness among genotypes. In other words, there's no favoring of superior traits. Basically, all five of these are completely unrealistic. In the real world, you never ever meet any of these conditions by and large. Sometimes you might have this one met if something's really isolated, if a population is isolated. But the rest of this stuff, you just generally, maybe this one will be met too, large population size. But the other ones, no. Mating is almost never random. Mutations happen all the time, and different genes offer different survival benefits depending on the environment. Which means the Hardy-Weinberg principle is pretty much never actually going to be observed. Or I should say, these conditions of the Hardy-Weinberg principle are never met. That's the better way to put it. These conditions are never actually met, which means evolution does occur. If all of these conditions were met, evolution would not occur. Gene frequencies would stay the same. Since most of these never get met in reality, most of these conditions never happen, evolution actually does occur because gene frequencies are going to change as a result of the opposite of these things. Non-random mating causes evolution. Gene flow causes evolution. Mutations cause evolution. Small population sizes leads to bottlenecks, genetic drift, among other things, causes evolution. And unequal fitness, which forces natural selection to choose some, causes evolution. So pretty much, these are never met. It can be a bit confusing, I understand. But that's the general idea of Hardy-Weinberg. So natural selection does happen, which means you have three general trends of selection on phenotypes. The first one is called stabilizing selection. So if you've got a particular phenotype, instead of natural selection favoring the biggest or the smallest, it tends to favor intermediate averages. One example for this would be human infant size. Babies that are very large when born can cause a lot of health issues potentially for the mother, but babies that are too small can have a lot of health issues for the infant. And so human infant size over time has favored that intermediate or average phenotype. I believe around 5 to 7 pounds is the average human baby weight. Directional selection tends to favor one extreme. One example of this is called island gigantism. The giant tortoises on the Galapagos are an example of this. And so when you have a predator-free environment, natural selection no longer is guiding toward protective measures, which means being big and energy efficient gets selected for, and organisms on islands tend to grow larger than the ancestral relatives on the mainland were that gave rise to them. Disruptive selection is when you tend to have opposite ends be favored while the intermediate is not. Basically, you have to pick a side of the fence. An example of this is in pepper moths, which I don't think you can see very well. So, the story of these guys, you may have heard it in a previous science class. They live on trees, and they live in trees, birch trees, I believe, in England. And you have the light form and the dark form. The light form was dominant pre-industrial revolution. All of the soot from industry darkened the trees, and so the dark form was actually better suited for that environment, and so they became more dominant. And as we cleaned up industry over time, pollution decreased. The light form has tended to increase in number as well. The light form camouflages well on tree bark like we have here with this, what I believe is a birch tree. It might be a different species of tree, but it's a tree with light bark. And so it camouflages well. The dark colored moth does not camouflage nearly as well. But if you have a mix of both of these, you're generally not going to do as well as either one, because there are trees as well with darker bark that our dark pepper moth can also survive in. So natural selection tends to favor either the dark or the light morph. There really doesn't seem to be much in favor of an intermediate, because, like I said, tree bark tends to either be this lighter color or it tends to be a darker color, which allows them to camouflage. Last topic for this lecture is genetic drift. Genetic drift is a change in allele frequencies due to random chance. Basically, some organisms get lucky and survive while others don't. This is a general example of how it would work. You got a small population of beetles, you got the shoe stepping on some of them. Unfortunately, the shoe happened to step where the green beetles were, so they died. And the tan beetles, they stayed pretty much unaffected by that environmental disaster on the beetle population. So this tends to affect small populations, because if a population is small, a random event can annihilate the population or annihilate a huge chunk of the population and to make a big dent in the genetic diversity of that population. For instance, let's see if I can use an illustrative example. Pretend that each English letter up here represents a particular version of a gene. We're just going to go out to E. Here's a large population. Here's a small population. They have the same genetic diversity, so to speak, because they have the 5 different versions of a particular allele. But over here, let's say there's a flood that wipes out a good chunk of individuals. If you look, A, B, C, D, E, still there. All all versions of the gene are still intact. A, B, C, D, E. Over here we have a flood that impacts them as well. We have just eliminated A, B, and C from the gene pool. And so even if this population replenishes, genetically it is less diverse, and that tends to be harmful for long-term survival of the species. So genetic drift tends to affect small populations because you simply have a greater chance of eliminating a particular version of a gene from the population if there's less of it present due to fewer individuals. So that's genetic drift.